Tissue succulence is typically defined as the ability of a plant to store substantial amounts of water in its roots, stems, or leaves. And the modern definition of tissue succulence is that the plant is able to store a certain amount of utilizable water so that it can withstand long periods of time without external water supply. I'd like to show you an example of just how thick succulent leaves can be. So this is a tobacco leaf, and this is a C3 non-succulent plant. And if we cut this leaf open, you can see that that leaf is very thin. So by comparison, let's look at another example of a leaf succulent. This is a kalanchoe leaf, and this is a Kim succulent, a leaf succulent, and you can see how thick that leaf is by comparison. Tissue succulence is associated with a number of other traits. The plants will typically have a thick cuticle, they'll have a low stomatal density, they'll have an abundant accumulation of epicuticular wax, and this causes the plant to limit its water loss to transpiration. And also, you'll find that the plants have highly responsive stomata, but also they will have shallow roots that in a rain will very quickly be able to take up and absorb water, and then when it's dry, they'll be able to limit water loss. Tissue succulence is very important for a plant because that stored water allows the plant to maintain its water potential and maintain active photosynthesis for long periods of time without any external water. And this adaptation allows plants to live or thrive in very arid or semi-arid regions throughout the world. Tissue succulence is typically associated with plants that have very large cells, but also have an ability to store water either in chlorophyllous tissue, meaning photosynthetically active, or non-chlorophyllous tissue. This tissue type usually does not have any chloroplasts or chlorophyll in it, and this is also referred to as a hydranchoma. In comparison to leaf succulents, we also have stem succulents, and this is an example of a stem succulent. And so this is a dragon fruit, and there you can see the stem, which has this inner white hydranchoma region with an outer chlorophyllous region where photosynthesis largely takes place. Tissue succulence occurs in about 300 different plant families. And if you look at all of the higher plant species, vascular plant species, it accounts for about 4% of all of those plants. Tissue succulence is associated with all different forms of photosynthesis. We have C3 photosynthesis, C4 photosynthesis, and crassulacean acid metabolism. Crassulacean acid metabolism, or CAM, is a water-saving type of photosynthesis that's often associated with tissue succulents, but it's not exclusive to tissue succulent plants. It's thought that tissue succulents evolved about 50 to 30 million years ago, and the adaptation really caught hold, and there was a large irradiation of different succulent species about 10 to 8 million years ago. About 15 to 8 million years ago, there was a decline in atmospheric CO2 levels, and this would have caused a stress on plants and would actually cause them to lose more water because they would have to keep their stomata open. And if a plant were tissue succulent, it would have an adaptive advantage under those conditions. And it's thought that that change in atmospheric CO2 level was one of the drivers that helped these succulent plants adapt and really thrive in increasingly arid environments. Succulent plants are found all over the globe, but they're particularly prominent in areas that are arid or semi-arid or have a dry season. And the Biodiversity hotspots are typically found in Central America or Mesoamerica, the eastern coast of Brazil, the tropical horn of Africa, southern Madagascar, and South Africa. There are many economically important succulent species. So this is Agave Tequilana. This is the plant from which tequila is made. The core of the plant, or piña, because it resembles a pineapple, is roasted and then it's fermented and then double distilled to obtain tequila. This is aloe vera. This plant has very important economic value. And this is used in the cosmetics industry. That's its primary use. That exudate is used in the cosmetic industry in many different formulations, and it's useful for wound healing and soothing burns. Here we have vanilla, the source of commercial vanilla, Vanilla planifolia, and this is an orchid species. When this plant flowers and it produces fruit, the dried seed pods are used for the production of commercial vanilla. This is Apuntia ficus indica. This plant has no true leaves at this stage in its development. These are actually modified stem segments, and they are called pads or cladodes. And if we can just take one of these off, so we just cut this pad in half, and then if you look at the tissue succulents here, we have this inner white layer. This is the hydranchema, and then an outer layer of chloranchema or chloroplast containing cells, and this is where the active photosynthesis occurs on the outer surface of the pad. This is an example of a succulent plant that is probably the most economically important in the world. Obviously this is pineapple, a baby one, and this is Ananas camosus, and the fruit is eaten throughout the world. It's very popular, and it's probably the most economically important. Also cam crop, in addition to being a tissue succulent plant. We think that engineering tissue succulents is going to be able to provide us with the ability to improve the drought tolerance and the salinity tolerance of some of our major crop plants. And the reason that we think that is that we've been doing some preliminary research on model plant systems like Arabidopsis. And when we do this, we have been able to successfully engineer tissue succulents, which you can see here on the left side of this panel. We see a control leaf, and on the right side we see a leaf which is much thicker, and it's about 40% thicker, and it contains up to 3 times more succulents and about 2 times more water content.
We test a variety of transcription factors from various plants, but the basic helix loop helix transcription factor from wine grapes was very useful for this task. This VHLH transcription factor is called CEV1, and it controls the process of cell size enlargement in the wine grapes berries, and is called engagement. And when we transferred to the model plant Arabidopsis, we found that all cell size were larger as in the succulent plant. Also decreases the intracellular airspace within the leaf, which presents a possibility that the CAM photosynthesis can be more successful in these plants. The CEV1 transcription factor overexpressing plants show a much greater availability to survive to water deficit stress and salinity stress conditions.
In addition to the larger cells throughout the plant, one of the other added benefits is that because these plants had more water content, they were able to become more drought tolerant or water deficit stress tolerant. And as you can see on this panel, the plants on the left side of each pot are control plants, and after an acute water stress of 15 days and then a recovery period all of those control plants died. And then on the right side of each plot, you can see our succulents engineered plants. All of those, or a much higher proportion of those, greater than 90%, were able to survive this acute water deficit stress. And the idea is that we could move this strategy to a major crop plant like soybean. We should be able to show that these engineered soybean plants will have more drought tolerance. The other added benefit that we didn't necessarily anticipate is that if you have a greater water content within the leaves of your plants, this presents the possibility that those plants could take up and dilute the salt and make the plants more salt tolerant. And what we see on this panel is a series of control plants, again on the left, and our succulents engineered plants on the right, irrigated with either 250 or 300 millimolar sodium chloride for 1 month. And you can see that the plants that had more water content were much more likely to survive.